Hi everybody, thanks for coming. Thank you IndieBio for the opportunity to present. My name's Ed Painter. I'm co-founder and CEO of A2A Pharmaceuticals. We're using supervised machine learning to create better therapeutics. The problem we're after is the pharmaceutical industry's repetitive failures in clinical trials due to efficacy and toxicity problems due to the overuse of tools like high-throughput screening for the same databases over and over. A2A Pharmaceuticals is doing something different. Similar to the design of object-oriented software, one line at a time and in individual modules, we're designing new molecules for the patients who need them, one element at a time in individual fragments. This journey started with the work of Dr. Matthew Welsh at Columbia University, where he was successful in developing the best new molecule for the RAS oncology target. This was used to form KaiRAS Therapeutics with a $5 million investment from Versant Ventures. The same technology was used to develop a new pharmaceutical for toxoplasmosis, 90 times more effective than the current application. This is with decades of industry effort and millions of dollars spent. The effort for toxoplasmosis took a matter of months. This video shows the Sculpt system at work. First, we designed very large databases with target-specific molecules, and then screen through those molecules for the ones with the most efficacious binding activity to the specific target for synthesis in the laboratory. In contrast with our competition, we're developing molecules from scratch with very small fragments. While most of our competition is going through existing databases that are relatively small, we develop databases as many as 100 million different molecules using supervised machine learning to analyze and pick the best ones. A2A Pharmaceuticals is a therapeutics company. The best way that we can leverage our technology is to develop medications for the patients that need them. In the case of mixed lineage leukemia, infants would be the primary beneficiary. Using the A2A technology Sculpt, with an iterative process, we were able to develop new molecules for synthesis, 7 of which we sent to Sanford Prebys in San Diego for analysis. Today, I'm happy to announce the results from those tests were incredible. Versus the competition from the University of Michigan, we were at least 2 times more efficacious. As you can see from the top row, it is very specific to the type of cell line that it's intended, with the cells not dying, except for those with the ones with the mixed lineage leukemia MLL menin disruption. We killed 97% of the cells in the dosage indicated here. We used the same process across 4 different cell lines, and were successful in every single one of those. This process was completed in just 4 months with the help of IndieBio and less than $100,000. This is despite industry efforts. Thank you. Comparatively, industry efforts took many, many years, at least 4 years and millions of dollars, and we won. So far, anyway. This is A2A's pipeline. I already told you about the leukemia drug. We're also focusing on a solid tumor indication, YAPTED, which is expressed across 50% on average of solid tumors, including pancreatic, lung, and liver cancer. This market could be well over $5 billion. All of our indications are likely to be over $1 billion. We're also focused on drug-resistant bacterial infections with gram-negative bacteria in broad indications across a number of different types of illnesses and tuberculosis. We've already generated revenues from partnerships with pharmaceutical companies. We're actively negotiating for our oncology programs right now with several different large pharmaceutical companies. Our business model will enable us ultimately to work on as many as 15 or more programs, spending less than $1 million per program on average, with potential proceeds of $400 million upfront and as much as $1 billion over the lifetime of an individual program. We're filing provisional patents on all of our programs, both on the algorithms and the data used to discover the new chemical entities as well as the new chemical entities themselves. I'm proud to be working with Dr. Matthew Welsh, who founded the Sculpt design, as well as Brendan Kelly from Stanford University, who has 7 years experience working with companies similar like Pfizer and Eli Lilly. Dr. Sridhar Vempati was responsible for identifying the specific targets in terms of their biological importance, as well as their commercial importance. Recently, Dr. Sturgiopoulos joined us as chairman of the board. He comes to us with 10 years of experience from Baxalta, Celgene, as well as, what's the other one, Novartis. He will help us with partnerships and has been very active already. Please join us at A2A Pharmaceuticals. We're raising $5 million. We're already fully subscribed. We're looking for additional strategic investors to get the new medications to the patients who need them. Thank you very much.